Good morning everyone. Welcome to all of you. In this video, we will discuss about the different types of exception of law of inheritance. That means the exception of law of dominance and exception of law of independent assortment. Because as we know that we have already discussed about the different name of the law of dominance and their exceptions. And as we also known that the law of segregation don't have any proper exceptions. So in this video we will discuss about the exception of law of dominance. And exception of law of dominance have many exceptions like incomplete dominance, codominance, lethality, epistatis, and many more. But we will discuss about the incomplete and codominance first. Okay, let me tell you about the incomplete dominance. Here the clarity comes from the name of the heading, that means incomplete or dominance. Incomplete dominance, and we are already known as about the dominance, that means each dominantial gene having the capacity to suppress the other genotype or other gene, that is called as dominance. And this means incomplete dominance means not fully suppressed to the other gene, that is called as incomplete. And it also known as intermediate or partial inheritance, or it also known as mosaic inheritance. Now look at some important points related to the incomplete dominance. It is the condition when gene of heterozygous condition are independent to each other. That means they are not dominant to each other, they are independent to each other. They are suppress their character by themselves. That is, when both alleles are present together, a new phenotypic discovered or new phenotypic is formed. And it was discovered by Codominance. Look at some examples of the incomplete dominance. The most and important examples are the 4 o'clock plant, that means the color of 4 plant. The color, it is the name of 4 o'clock plant, that is Mirabilis jalapa. 4 o'clock means it is the flower which opens at the time of 4 o'clock, that's why it's named as a 4 o'clock plant. And the flower of snapdragon, or it is also called as dog flower, because their mouth is just look like as the dog face. This is some examples of the incomplete dominance. Look at here, we will also discuss about the example. Just look at here, it is the example of 4 o'clock plant, and we are putting here the pure line red flower. Pure line red flower. What is the meaning of pure line? As I told you earlier, that is the pure line, either dominant or either recessive. That means homozygous recessive or homozygous dominant is also called as pure line. It is the pure line. That means, or it is also impure line. Two different types of genome. We are putting here about the pure line red flower and the pure line white flower. Both are homozygous condition. So here the genotypic will be red, for red color RR, and for white color small r and small r. We have already discussed about in the law of dominance, the crossing over of these two gametes would be red. But here the capital R is not dominant over R, and it shows their equal dominance to each other, that means capital R and small r, between capital R and small r. That's why it gives a different and new type of phenotypic, that is impure pure line of pink color flower. Here the produces no red color, no white color. The new phenotypic is produced or formed in the form of like pure pink color. Are you understanding the point? That is, the incomplete dominance means they do not carry their parental generation or parental character, that means red color or white color, that is form a new phenotypic, that is pink color in F1 generation. Well, what would happen in the dominancy? It gives the dominance to the small r and it gives the red in flower. But here it is not happens. That's why it give a pink color flower. Is it clear? And what will about when the selfing will happen? When the selfing occurs between same type of gene, that is the capital R and small r, capital R and small r, and as you know that it will produce the two different types of gamete, and it also produce two different types of gamete, and here the some crosses will happen in this format. You can also use a Punnett square box. In the Punnett square box, here the capital R and here small r, here also capital R and small r. It will be capital R, it will be capital R, small r, capital R, small r, and small r, small r. That means one is pure line flower carried just as their before generation, that means the F1 generations, parental generations, because it is F1 generation and it is F2 generation, and this is the capital R and capital R, that is parental generation, and this is the small r and small r, it is also parental generation. But this is the two different types of phenotypic produce which is comes from the F1 generation. Is it clear? That means, can you tell me what will about the genotypic and phenotypic ratio for the incomplete dominance? If you are saying the 3:1 phenotypic and genotypic 1:2:1, it is wrong, because here it is a red color, it is a white color, and this is the pink color. That means the genotypic also different for the different color and phenotypic is also different for the different characteristics. That's why what will be the resultant and what will be the result? The result will be genotypic plus phenotypic, or genotypic or pheno, both will be 1:2:1. Is it clear? Genotypic, why? Because you can already see here capital R is 1, capital R small r 2, and small r 1. And phenotypic, that means the external appearance. External appearance is red in color, this is the pink in color 2, and white in color. Is it okay? That's why it does not obey the rule of law of dominance. That's why it's categorized in the exception of law of dominance as the incomplete dominance. Okay, now look at here about the different types of exception of law of dominance, that is codominance. Co means equal and dominance means dominancy. Each gene or each allele dominate towards each other, that means the equal effective on both the genes. Look at here some important, and it will be clear for you. The both the allele which are present together in heterozygous condition but neither show dominancy recessive nor show neither intermediate condition but express their traits independently as known as codominance. That means both character will produce, suppose it is a T and T, it is not radical tall plant and it is not different types of plant. That means it will be also effective on same That means 50% effective for capital T and 50% effective for the small t. Equal dominancy to each other. It will be effective on this and it will be effective on capital T. That is called as codominance. And the most common example for the codominance is coat color of cattle, AB blood group, coat color of mice, and MN group of blood human group. We will discuss each and every points. Firstly, we will see about the coat color of cattle, how it works for the codominance. Codominance. Look at here about the coat color of cattle. That means the skin color of cattle. We are taking here the white coat color of bull and it crossed with the black coat color of cow. When it is, I think this is not time to show their genotypic condition and their gametic formation, because you have already seen many more time, so we will understand this. This is a genotypic formation capital W, and this is for coat color of black coat color of cow, small w and small w, and this will be the reaction. That means capital W and small w. Is it clear? And look at here about it will be produces not red color, sorry, not white color, not black color. That means equal effective color, black and white color. Codominance, equal effective on each other. That is called as codominance. The F1 generation will be seen in the black and white. The some character of black skin will happen in the F1 generation, and the some color of the black cow will also present on the F1 generation. Black and white, both the color will formed in F1 generation, their progeny. Is it clear? Now look at here about the F2 generation. It is the F1 generation, as I told you, and it is the equal effective to each other. And what will happen when the selfing will start? Selfing. The selfing means the two same type of F1 generation will cross together. Capital, it will produces two different gametes, that is a reduction, and it also produces two different gamete. And look at here about the products of F2 generation. It is the F2 generation. F2 generation. This is the male, that is bull, and this is female, that is cow. It produces the capital W and small w, and capital W cross with capital W, that will produces both capital W. That is, what is the phenotypic of this condition? This is the white coat color, and this is black and white, and this is black and white also, and this is black coat color. Is it clear? That means it also shown the homo white color. That means homozygous white color. Homozygous white color, that is 1. Hetero black and white color, this is black and white coat color, is 2, and homo black coat color is 1. That's why the phenotypic and genotypic ratio for the codominance of coat color of mice is 1:2:1. Is it clear? That means the phenotypic also will 1:2:1, and genotypic also will 1:2:1 for the codominance. Is it clear? Any doubt between codominance and incomplete dominance? No. Look at here about the other example of codominance, that is the ABO blood group, which is found in human. That is blood group A, blood group B, O, and AB. These most of the blood group are found in a human. What is the blood group? The group of antibody and antigen in the blood of a human is called blood group. It is discovered by Landsteiner and Wiener. 4 types of blood group is found in a human, and let's see about the different blood group types. Blood group having the A blood group B, blood group AB and blood group O. Each and every blood group having present the antigen and antibody both. But the blood group A have antigen A present in the blood group A and antibody will be B. The nature will be the normal in condition. It is as same as for the blood group B also. The blood group B having the antigen B and antibody A, and it also be a normal condition. While for blood group AB, the antigen, both the antigen will be present, that means A and B, while the antibody will be absent in AB blood group. That's why it is called as universal receptor, because it don't have any kind of antibody. That means it do not donate the any type of blood group. Look at here about the blood group O. Blood group have absent of antigen, while the antibody will be, both antibody A and B will found. That's why it is called as universal donor. Are you getting my point? The blood group O and blood group AB is the most important blood group, because this is the universal receptor, while the blood group A is universal donor, because blood group O having the both antibody A and B is present in the blood group O. Is it clear? Now look at here some important point related to the blood groups. Out of 4 blood group, the blood group B is more abundant than the human. That is, the blood group B type of human mostly present, and that is it is all about the 60% of blood group B is present in human. The inheritance of blood group. The inheritance of blood group represented by I gene. And I gene for the blood group always represented is multiple allele. Blood group ABO, it shows the multiple allele. Look at here the inheritance of I gene, that is it is of 3 types: IA, IB and IO, and it also represented by small I. Look at here, it will be clearly more about the blood group. It is the blood group A, that means it is a inheritance of A, and B and IO. IA always dominance over IO. That means IA, that means it will be produces a homozygous and heterozygous condition for blood group A, and it will be always dominant over O. And same for the IB. IB is also dominant over the IO always, but IA and IB always co-dominance to each other. That means A will not suppress to the B and B will not suppress to the A. That will co-affected. That's what is called as codominance. Look at here some important phenotypic blood group and their genotypes. Blood group A carry 2 genotype. That means the homozygous condition, it is the homozygous condition, while it is heterozygous condition. This is homozygous condition. Homozygous genotype and heterozygous. That means, as you know, that it is always present in a pair form. Allele is always present in a pair form, that is IA and IA produces the blood group A, and IO also produces the blood group O. Why? Because I told you IA always dominant over IO, that why it gives their dominancy form, that why it produces the blood group A. Just as blood group A, it is also applicable for the blood group B. That means B, it is homozygous condition and it is heterozygous condition. And blood group AB As I told you, it is only a blood group which have only both the blood group A and blood group B inheritance or condition will present in this form. That's why IA and IB is present. That is heterozygous. It will show always heterozygous condition. And O always show homozygous condition. That is either IO or small I. This is the 6th type of different genotypes of different blood groups. Look at here some important point and important term. The number of phenotype will be 4 phenotype: blood group A, blood group B, AB and O. The number of genotype will be 6. That means 1, 2, 3, 4, 5 and 6. And number of heterozygous gene pair. Heterozygous. This is the heterozygous condition 1. This is the heterozygous condition 2. And this is the heterozygous condition 3. That means 1, 2 and 3. This is the heterozygous gene pair condition. Pair and heterozygous. That is 3. And we will also use a simple formula for the multiple allele and for the number of genotype. When the some question will be comes in the form of the how many number of gametes will produces in multiple allele type of question. This is the simple formula for that. The number of genotype is equal to n into n plus by 1. This is the formula by 2. And number of heterozygous pair, n into n minus 1 by 2. This is the where n is number of allele. Number of allele. This is the simple formula for what? For their multiple alleles. So multiple allele. Look at here. I'm giving to you 2 questions. You will have to make the answer on your answer sheet, and we will also describe what their answer, but you have to try first. So the first question is a male with heterozygous blood group A. A male with heterozygous blood group A marries with a female the blood group AB. That means the female having blood group AB and male having blood group heterozygous condition A. And what will be the type of offspring and what will be the product and what will be the type of blood group? What will be the type of blood group? This is the first question, and this is the second question. If 6 gene or 6 allele represented in 6 allele, that is A, B, B, C, C, A, C, D, E and F. That is the 6 different allele. 6 different allele represented as a single allele, single set. Then what will be the number of genotype and what will be the number of heterozygous gene pair in this question? This is a 2 different question. You will have to make this answer in your notebook. For this question, I will also solve this problem right here, right now, but you have to try first. So you can pause this video, and after the making your answer, match the answer which I made making here. Okay. Look at here. The first question is for the male is heterozygous A blood group. That is the heterozygous condition. And female, look at here, female and male. It is male and female. Male is heterozygous A and female is AB. AB. What will be the product? This will produces 2 different types of gamete, that is IA and IO, and it produces 2 gametes, that is IA and IB, and the crosses will produces between IA and IA, that is IA and IA, and also this IA crossed with IB and it produces IA and IB, and IB is IO. This is IA and IO, and this is also for IB and IO. This is the four different types. This is the homozygous condition for blood group A. This is heterozygous condition and is also and only it is applicable for the blood group AB, A and B, AB, and this is heterozygous blood group A, and this is heterozygous blood group B. These four types of blood group will found in their offspring. Is it clear? The blood group A, blood group B, and blood group AB. Here also the question is rising from the percentage of the blood group, different blood groups. So what will be the percentage? The percentage for the blood group A is 50%, because 2 out of 4 is what? 2 out of 4 is A blood group. That's why it is shows at 50%, and AB is 24% because it is produces 1 by 4, and for blood group B is also 1 by 4. That means it is also 25% and it will be 25%. Is it clear? This is the question number first. For the heterozygous condition for male and female, this will be the product. And what will about the question number 2? What is the question number 2? Question number 2 is we have to make the formula first. What is the formula for the number of genotype? The formula is number of genotype. Formula is n into n plus 1 by 2. That means n is equal to number of allele, multiple allele. The 6 is represented as a 1 allele. That means 6 into 6 plus 1 by 2. 6 into 7 by 2. It is very simple, and it is 3. That means 21. 21 will be number of genotype. And what about the gene pair? N into n minus 1 by 2. That means 6 into 6 minus 1 by 2. 6 into 5 by 2, or it is 3. That means 15 gene pair will be produces in this question. Is it clear? Here the solution of the given before the question. It is the first answer of the first question and it is the second answer for the second question. The number of genotype will be produced 21 and number of gene pair produces 15. Any doubt? This was the exception of law of dominance, that is the codominance and incomplete dominance. Now we will also discuss about the exception of law of independent assortment, that is linkage. We will discuss in the next video. For that, keep watching and stay tuned. Take care and bye-bye.